Okay, it's 11 o'clock. Good morning, a happy Sunday, and welcome to yet another online Chai and Wine. It's November, and we continue with our Chai and Wine sessions celebrating the Nobel Prizes for the year. This week, it's the Chemistry Prize, which has been given to this gene editing technique called CRISPR. That sounds very crisp. And to tell us about it are two young biologists, Mahima and Tuli. They're both from Shubha Tole's lab at TIFR, where Tuli just finished with a master's and is actually sitting at home in Kolkata right now. Mahima is probably sitting in the lab in Mumbai, and she works on trying to understand how different cells in the brain take on different identities. Mahima also says that she is a Sherlock Holmes fan. And Tuli, of course, has now, given that she has a little break after graduating, set up a biology web magazine with another student, Asmita, and it's called Wide Angle. You can see the link in the YouTube description later on. And she likes poetry and publishes a lot of them here and there as well. But today it's going to be time to talk about CRISPR and biology. So without further ado, let's go over. It's up to you, and we're all set and ready. Okay, so a reminder to the online audience, please do post your questions as comments in the chat window on YouTube or on Facebook, wherever you're listening. We will try and take them either during the session or at the end after they finish the talk. So keep them coming and we'll try and answer them as well. We also have Varun from the Department of Biological Sciences here, who will also help in coordinating and asking questions. Meanwhile, if there is anything, if you cannot hear them, cannot see clearly, please put comments on YouTube, because unlike a live audience, there is no other way we have to get feedback from you. So do send us your comments. Okay, let's get started.
Hello, am I audible?
Yes.
Hello.
Yeah, Tuli.
Yeah, go ahead. Go ahead. You guys are audible.
Okay. Okay. Hello, everyone. Thank you for joining us on this Sunday. And okay. Okay. Hello, everyone. Thank you for joining us today on this Sunday. And it gives me immense pleasure to bring to you the Chai and Wine lecture on this year's Nobel Prize in Chemistry. So this year, the prize was given to Jennifer Doudna and Emma Charpentier. They became the sixth and the seventh women in the history of Nobel Prizes given in chemistry. So I'm glad to present their work alongside my friend and colleague Mahima Bose on how CRISPR became the phenomenon that it is today. Okay. So to start with their journey,
So, Tuli, hi everyone. I'm Mahima and I will be co-hosting with Tuli. And this year's Nobel Prize is given to Jennifer Doudna and Charpentier. And this year we are both are presenting it. So, Tuli, why don't you tell us a bit about the journey of these two scientists into the field of biology and how they actually went about to discover CRISPR?
yes, sure. Can we have the slide, please? Yep. So here is Professor Jennifer Doudna. She was born in Washington, USA, and she did her PhD from the Harvard University and is currently a professor in the University of California in Berkeley, where she pioneered this CRISPR-Cas9 technique.
On the other hand, Emmanuelle Charpentier was born in France. She did her PhD in microbiology from the very famous Institute of Pasteur in France. And she went to join many other institutes before she actually landed in UMIA Center of Microbial Research. This is the place where she actually worked on this Nobel Prize winning discovery. And that's how this is where she actually did most of her work. She currently is in Max Planck, but Jennifer Doudna and Emmanuelle Charpentier had a long journey. They actually were working on something similar, and they met at a conference in Puerto Rico where they shook hands to do a collaborative work, and that work finally led them to this Nobel Prize winning discovery.
Okay, so CRISPR has been one of the most talked about discovery in the modern times because it has revolutionized the way we are able to treat medical health issues and agricultural issues. As well as somewhere in between, scientists are also trying to make monkeys that are smarter. Well, the last part was an extrapolation, but let us start at the beginning so that we can understand better why CRISPR is such a phenomenon.
Well, CRISPR has been told to be a gene editing tool. And to understand what gene editing means and what it can do to life on Earth, let's understand what life actually is. So, in this planet, we have various kinds of organisms coming from single cell bacteria to much bigger, complex, and broader spectrum of organisms. And one of them is humans. We are humans, but all of them have something in common. They are all made up of the building blocks called the cells. The cell in itself is a world, and inside the cell is something called the DNA. Now, you have heard of DNA, but do you know what it actually is comprised of? So, if we take a closer look into the structure of DNA, it is like a ladder. It has two parts to it, and it has building blocks in itself. And those have there are actually four building blocks. We call them A, T, G, and C. There's a tendency in the DNA that the A has a very strong bond with T. So they are like the best friends. They cannot pair bond with anyone else. And G has a strong bond with another molecule, C. And this is the basis of all the understanding. This comprises of the long convoluted code, which is the DNA. And this code actually tells how humans look, how they, for example, one of the features is the eye color. So how your eyes color is as actually determined by the code present in your DNA. It can be blue, some people have gray eyes, some people have green eyes, some people have brown eyes. And that actually comes from the DNA that you have in your cells. But how does DNA work, and how do you get all these features? So imagine there's a city, and inside the city there's a particular area, and that is the palace where the ruler resides. A similar thing happens in the cell, which is itself a city, and inside that is a particular small, very composed area called the nucleus, where the DNA resides. Now, the king actually sends out messengers, and similarly, the DNA that they follow the king's orders. And what the messenger does is it goes outside the city and it recruits workers. And these workers do all the job the king wants the cell to do. And similar thing happens inside the cell where the RNA is actually recruiting or producing certain things called proteins. And these proteins do all the job inside the cell, from gatekeeping as to who might enter and not enter the cell, when to destroy the cell when it's in danger, how to transport stuff from one part of the cell to other, everything. So it's a city in itself, and proteins do all the work. And that's what happens inside the cell. And so it's an instruction manual, but there's another part to it. The king also has a job to reproduce itself. So DNA has to make more of itself so that the monarchy continues. And that's what happens inside the cell. Now, Tuli, do you think this all this job is foolproof? And does this happen without any faults or errors?
Hey, Mahima. Of course not. You see, we are what our genes dictate us, like you said, and genes are nothing but the stretches of DNA. And normally, a gene would make a protein via the guidance of the RNA, and that protein would be functional and do all the job, like you said. However, sometimes there might be an alteration in these genes, which may lead to a protein being abnormal or no protein being formed at all. So, at the base of most our diseases.
Wait, Tuli, hold on a second. I think I'm unable to.
Share your screen again. No
Yeah,
worries.
I'm sharing. Yeah.
Okay, so like I was saying, a normal gene would give rise to a normal protein, which would do all the job in the cell, right? However, sometimes this normal gene can get altered into a mutated gene, as a result of which the protein that is produced also is abnormal, or sometimes there is no protein produced at all. So at the base of of most our human diseases are these gene mutations, right? So this is where you can understand how important it will be if we had a tool that could actually remove these mutations so that things can go back to normal. And this is where gene editing comes in. Now let me give you a sense of how difficult this problem is. So imagine this manual as a book, right? And the book is as big as, say, for example, the Harry Potter and the Order of the Phoenix. Okay, it's a big book. And somewhere in this book are these mistakes, like the one here, which says hit the head on the nail, or something like do not eat inside the laboratory. So clearly, these sentences make no sense. What the actual instruction should have been is hit the nail on the head or do not eat inside the laboratory. So when such things happen within the cell, they lead to diseases, as a result of which CRISPR becomes very important, because CRISPR can do this entire scanning of the entire genome with precision and within a short period of time. So let us now embark on a journey to understand how CRISPR became this powerful tool of gene editing. So the first person to stumble upon CRISPR was Ishino, and he was simply analyzing the genome of a particular bacteria. And there he found these really peculiar sequences, which were repeating not twice, not thrice, but up to 5 times. And he was wondering, what are these sequences? Why are they here? But he didn't have the tools to look at them further, but he reported it to the community anyway. A few years later came the person, Mojica, and he was staying at this beautiful beach city of Alicante in Spain and studying how this particular bacteria lives in the salty water of the sea. And there too, surprisingly, he found the same sequences, repeated one after the other. So, what were these sequences that they were looking at? They were actually short repeats of short repeating sequences that were of palindromic in nature. So, what are palindromic sequences? They are just strings of letters that, when read from front or from the back, you would get the same word. For example, Malayalam. You read it from left to right or from right to left, you get Malayalam. For the gene within the DNA, a sequence like this would be a palindromic sequence, which if you read on the top would read AATT, and when you read it from the bottom, it also reads AATT. So these repeating units are nothing but made up of such palindromic sequences. In between these repeating units are actually some uh, spacer DNAs, and so the name came clustered, regularly interspaced short palindromic repeats. Clustered because they occur within a particular region of the DNA, and because they are spaced by these spacer sequences, and since they are palindromic repeats, and so we get the well-known acronym CRISPR. And both Mojica and the scientist Janssen, their groups agreed to stick to this name, and that is the name that got carried forward in the field. Janssen also found these genes very close to these CRISPR array, and he called them simply CRISPR associated genes, or what we know today as the Cas genes. But in spite of having all this information about the genes and the sequences, the field did not know what were they doing in the bacteria. Why did the bacteria need them at all? So, Mahima, can you tell us what were the sequences and why were they needed?
Good question, Tuli. For all these years, they figured out the sequences, they figured out the regions, but they actually could not figure out what they did. And after a lot of study by a lot of scientists, there was a clue on what these sequences actually might do. So it's not like a Sherlock Holmes episode where Sherlock is the only one who solves all the problems and comes out with a solution. In science, it's always a continuous process of discoveries, and it does not happen by just one person. There is no one single hero. It's a collaborative effort, and many people actually work together to find out answers to these problems that are existing. Something similar happened in the field of CRISPR. So people actually looked at these regions, and one by one, scientists were actually decode what these regions mean. So there are these repeat sequences, which we talked about. These are present in between the spacers. Now, people thought, what are these spacer regions? Because these spacer regions were different in all the bacteria. No two bacteria had the same spacer region. But what did they signify? And it came after, I mean, in 2005 or something, people actually figured out what these spacers mean. And it had to do with something the bacteria wants to itself. The bacteria has a goal, not like our goals where we want to get a PhD, want to do great science, want to succeed and earn a lot of money. Bacteria is simple. Bacteria has just one goal in life, and that goal is to stay alive or to produce more of itself. So what happens sometimes is the bacteria is attacked by some pathogens called the viruses. And these viruses are a big threat to bacteria because they might try to kill it, because the virus itself wants to stay alive. So what people found is these spacer regions are actually viral DNA particles. So the virus sometimes attacks the bacteria by injecting its genetic material into the bacterium. And a part of that viral bacterium is somehow gets coded inside the bacterial genome. So what the bacteria does is it creates a mugshot. some virus has come to attack the bacterium, and the bacterium has now remembered it that this is an attacker, this one is an intruder, and next time this one comes, I will remember and raise an alarm. And something similar happens, and the spacers which we see are actually parts of a viral genome that is a memory encoded into the bacterium of an attacker virus. People found out that these spacer regions code for something important. And here you can actually see that next time when the virus comes to attack the bacterium again, the memory region codes for an RNA molecule. And this RNA molecule has a very important role. Its sequence remembers the virus's sequence. Now, Tuli told you about the Cas genes. People found out the importance of the Cas genes. Now, Cas genes code for Cas proteins. The Cas proteins have various functions, but one of them, called the Cas9, has been shown to have features of nucleases. I mean, they can be used as scissors. So people speculated that this scissor molecule can work with the CRISPR molecule to actually help degrade the intruder DNA. But is it the entire picture? Turns out no. So you see, this is kind of a chemical reaction. There's a DNA, there is this protein, and there's this RNA. And the chemical reaction's end product is DNA being cut. So scientists asked that, can this be done outside the bacterium? Well, these findings are also, so that's what was the next question. And the finding that these Cas genes and CRISPR arrays have been a part of the immune system was found out by a group of scientists working independently, Dr. Mojica, Dr. Bolotin, and Porcel, who showed that CRISPR is a nucleic acid-based immune system. But the question was, can this be done outside the bacterium? So if you take a target DNA sequence, the Cas proteins and CRISPR RNA, will it be able to cut the DNA? Is this the entire thing needed for DNA cutting? Turns out no. So for years, people worked on this but couldn't figure out how this DNA is getting cleaved. What is the actual mechanism? Nobody could understand it. But then came the work of Emmanuel Charpentier, whose lab in 2010 or 11 actually figured out another piece in the puzzle, and that was tracer RNA. Now, what is this tracer RNA? So what they showed was, if they put tracer RNA into the cocktail, they could actually see the DNA being cleaved. So it's not just Cas9 and CRISPR RNA that are sufficient to cause the DNA to be cut. There's tracer RNA as well, which is an important player. But how is it working? So in the CRISPR array, there are these CRISPR genes associated with it, but there is also another region which is critical for its functioning. That's the tracer region. The CRISPR arrays does not produce a mature RNA in itself. They produce what is called an immature RNA portion, a pre-CRISPR RNA segment. And tracer RNA's role is to actually bind to these CRISPR RNA regions and create a mature CRISPR tracer complex. So tracer is actually causing the maturation of CRISPR RNAs. So now Emmanuel Charpentier had some idea how this might work, but it was still not clear. So at the conference in Puerto Rico, she met Jennifer Doudna. The question was whether this complex is the one that recruits Cas9, the nuclease, the scissor, to actually cut the DNA. Now, Jennifer Doudna was working on the structure of Cas proteins, and Emmanuel Charpentier has just published about this complex CRISPR RNA, tracer RNA.
Mahima, could you just repeat this? The earlier slide, you had this very nice thing about this tracer RNA, and can you just go through this once more, maybe? It's a little, yeah, from this tracer to this pre-RNA, the complex. Can you just repeat that part once more?
Yeah, okay. So this CRISPR RNA array, which has multiple regions of multiple viruses encoded in it, this does not come about and function on its own. It is encoded as a premature or a pre-CRISPR RNA copy. Now, tracer's job is to actually mature this complex into a functional complex that is actually capable of cutting the viral DNA genome. So, for example, if you look at this green portion, this encodes a region that the green colored virus put. So when tracer region binds to this and this thing matures into a complex, and how this happens is still not very clear, but this mature CRISPR RNA tracer RNA complex is somehow important for the cleavage of the viral genome. Is it clear now? Hello?
Yeah, thanks. Go ahead.
Yeah,
Go ahead.
okay. So I was saying that although there is this complex formation that is happening, but is this the complex that is recruiting the Cas9 scissors? That work was done in collaboration with Jennifer Doudna. Jennifer Doudna was looking at the protein Cas9, and Emmanuel Charpentier just worked on the RNA complexes. So they actually realized that the CRISPR RNA is not that simple. It has a region that binds to the viral genome that we understood, but it also has a region that is complementary to the tracer RNA part. So this part binds to the tracer RNA, and the other part actually recognizes the DNA segment of the intruder. Now, there is this another part called tracer RNA, which we talked about. Now, there are two components of a chemical reaction. In order to make this chemical reaction more efficient, what this group did was to create a single guide RNA molecule, which was a combination of the CRISPR RNA and tracer RNA complex. This actually led them to understand how these two function, and if these two are fused together, will they work the same way as CRISPR tracer complex does? And is this sufficient to bring about the reaction? And what they saw in bacterial DNA, that when this single guide RNA was put in the cocktail with Cas9, the enzyme that cuts, they could actually see DNA cleavage to happen, which showed that these three components are sufficient to cause the DNA to break and the process to happen.
Mahima, before you go ahead, we have a question from a me cool 25, which reads: Is tracer RNA same across all viruses?
Well, it depends on strain to strain. There are slight differences, but tracer RNA sequence overall is the same. The difference is the part of the CRISPR RNA that encodes for the intruder. So there might be slight differences, but more or less the sequence is the same. Okay. Okay. Wait,
At this time, I think there's one more pressing question that has just come up while you put your slide back. If this is the thing, uh, is CRRNA coded for how many viral genomes, or has it, or it has some particular sequences for all viruses?
So there's a particular region uh, in the tracer complex that detects an intruder. It's called a PAM sequence. So CRISPR sequence can be anything, depends on the intruder. And based on the PAM sequence, the machinery is able to detect that this is self and this is foreign. And that's how it's able to differentiate and cause a nick. And as far as as far as I understood, there can be n number of CRISPR arrays and many tracer RNAs complementary to it. I mean, tracer RNA is the same in one particular strain that binds to CRISPR arrays, but the sequence that is complementary to the part of the viral genome is different. Was that the answer?
Well, yes, carry on.
Okay. So many of us don't know this, but there's another scientist, Virginia Sikinsis, who published the same finding at the same year, just a few months later than Jennifer Doudna and Emmanuel Charpentier. And because of the peer review process, his publication got delayed. But he also actually proved the same thing independent of this duo. And that's why somehow his findings are overlooked. Now this led to now the discovery of that the entire mechanism of CRISPR RNA, tracer RNA, Cas proteins actually led to something very interesting, and that's what is in the news right now. It's the era of genome engineering. There were three groups in 2013 who independently tried to achieve something, and that was to replicate this entire system of CRISPR-Cas array from bacteria into eukaryotes, in for example human cell lines or something which don't have this CRISPR immune system, and to ask whether it works or not. And so they created a DNA code that will produce a guide RNA against a mammalian gene, a gene which bacteria does not have, but we have, and also a Cas protein. And they induced it into human cell lines to see whether it works or not. And what they saw was that this system also works in mammalian cell lines, and it was able to cut the human DNA and cause a successful double-stranded break. But the double-stranded break that happens for the viral DNA versus our DNA, is it the same thing? What happens if we cut human DNA? How does it respond? How does the cell respond to something like this? So what happens once the DNA is cut? Will we achieve what we want to achieve? To understand this, let me tell you some analogy. For example, if there's a patient if there's a person who went through an accident and some part of his body or a face got mutilated, and the surgeon's job is to repair the face and bring back to its original form. Now the surgeon can do to the best of her ability if she does not have any idea how the person actually used to look earlier. But if the surgeon had some reference, some photograph of that person, she might be able to do a better job at actually recreating the original face of the patient. Something similar happens in the cells of mammals, the eukaryotic system. There's a DNA repair machinery that acts like the surgeon when the patient, that is the DNA, undergoes some damage. So once the DNA is cut, the repair machinery comes up And if it does not have any reference in next to it, anything near its vicinity that actually tells how this strand actually was earlier, it tries to do a good job, but sometimes it does not actually recreate its original sequence, and this leads to mutations and some different sequence than the original one. And this part actually sometimes leads to gene mutations. And if this mutation happens, the gene can become non-functional. And that way, gene knockouts are created. So if you target CRISPR against a particular gene and cut it, the gene will fuse, but it might lose its function. And the other way, however, how the DNA repair machinery works is if it has a reference DNA just around it somewhere, the repair machinery is actually able to recreate the entire system to its full way. Now, how do we use this information? So what scientists actually did was hijack the repair machinery to add new genes. Now, we understood CRISPR is useful for removing genes, but can we achieve something else? Can we move a step further? Can we add new genes instead of just removing genes? So if we provide a reference DNA that is similar to the original sequence, the repair machinery can actually understand it as a reference and actually recreate it. But what scientists did was hijack this machinery and provided a reference, but a small part which is something extra, a new gene. So what the machinery thought was this extra part is also a part of this DNA segment. And in this way, new genes are added into the genome. So if you have a cell and you put the CRISPR machinery along with a DNA segment from outside that has parts which are similar but also has a part which is different, the cell actually understands it as the same thing and adds the new gene into the genome. So if you put some cell which codes for some new color or fluorescence, the cell might be start showing this new phenotype or new form, new color or new fluorescence within itself. And that's how scientists got successful in achieving this kind of genome engineering. Now this mechanism actually won many awards before the Nobel Prize itself. Jennifer Doudna, Sixinnis, and Charpentier won the Breakthrough of the Year award and also the prestigious Kavli Award in 2015 and 18 respectively for actually discovering this way of genome engineering. And another prestigious award, the Canada Gairdner Award, was provided to Doudna, Charpentier, and Feng Zhang from MIT for developing this technique and making it a step further on the ground of genome engineering.
Okay, 1 second before we move to the next part where CRISPR is changing the world. There's a question and it reads this: Is the trigger is there a trigger that enables the combining of the CRISPR RNA and the trigger RNA in the cell?
Is there a trigger?
Yes.
Okay, good question, actually. So these RNAs are continuously produced, and it depends on chance, actually. When these complex uh, get in, might interact with the intruder. So as far as I know, these don't have a trigger, but they are there in the cell, and they interact with the foreign material, and whenever they find a pairing, they cut it. Tuli, am I right? Do you know anything about the trigger part?
No, the entry of the viral genome itself acts as a trigger, and there are other Cas proteins that sort of act as a policeman here to inform that there is this intruder coming in, but not a trigger as such that I can think of. Yeah.
Okay. Now
Okay, thank
this
you.
now this story does not end here. We all know CRISPR has been changing the world. CRISPR technology has revolutionized the entire field of medicine, agriculture, therapeutics, and everything. There are various applications. People are actually working on how to use CRISPR in their field. One of them is agriculture. So people are using CRISPR to make plants pest resistant, like bananas and chocolates. They are being modified so that they don't have they have resistance against all these pests or insects that attack or make the plants go bad. And there are also new varieties of coffee and wine coming up, so that new for example, in coffee, people used to spend a lot of time and resources on making a coffee decaffeinated, but people are now changing the plant itself to produce decaffeinated coffee. And for wine itself, for wine, they are changing the grape plants to produce less sugar so that there is wine with low carbs. And that's how CRISPR is making all these changes in the field of agriculture. People might think that there have been these technologies and we have heard of GMOs, genetically modified plants, but CRISPR is a step ahead. The time needed for creating these modified plants was at least 10 years through the conventional methods. But if we use CRISPR, the time reduces to almost half. And that's how CRISPR has been promising gene editing tool in the field of agriculture.
So Mahima, by when can we expect Karela to stop tasting like Karela?
Yeah, I don't know. Will you work on it in your
Uh,
life?
that is a real
So,
struggle for a lot of people.
Okay,
so
speakers, before you move on with more CRISPR, there are two very good questions from a Swapnil Pradhan. And the question is as follows: what did people use uh, before this? That is, how is site-directed mutagenesis? How is this different than site-directed mutagenesis? Is this discovery fundamental or applied?
Tuli, will you like to answer this?
Uh, okay. Can I rather answer it at the end? Will that be okay?
Yeah, that will be
The
okay.
second part of the question, is this discovery fundamental or applied? Uh, well, I
It's a
think
mix of both.
it's a mix of both, but I would say that it's a fundamental thing because it's using something that has been present in nature for millions of years and transforming it, or rather applying it to do something that is of our interest. So yeah, it's a mixture of both.
Yeah. So, Tulil, would you like to tell us how human therapeutics field is being affected by the advent of CRISPR?
Yes, absolutely. So human therapeutics, what can CRISPR do here? So what can be done is that to make sure that certain the diseased cells of our body can be treated. The For example, if the disease is of the blood, you can actually take blood from the patient and you can treat it with CRISPR, as in you can edit its genome the way you want, and then you can return back these cells back to the patient. So what this does is that it overcomes the complications that can arise, the immune complications that can arise if, say, the edited blood edited blood cells would come from some other source, right? What can also be done is to put back edited to edit the genome of human patients. Is that the CRISPR-Cas9 system can be packaged within these friendly packets that are made up of lipids, okay, which will not have any immune response. And they can be returned back to the human through their blood so that it can be circulated and it can go act wherever it is supposed to, or it can also be returned targetedly to a particular organ like the liver where it is required, right? So this is human therapeutics. CRISPR is also now being used for, can I have a slide change, please? Hello? Hello? Hello, am I audible?
Yes, you are audible.
Oh, I'm audible, but I think the slide is missing.
Yeah, it's come
Yes.
back. It's come back. Carry on.
Okay. Thank you, Varun. Okay. Okay. So should I repeat this part? I don't know where I trailed off. Okay, I'll just start from here again. So like I was saying before, in case you didn't hear me, cells from a human patient can be taken out and treated with CRISPR-Cas9, and then they can be put back into the body of the human of that patient to overcome the immune complications that may arise if those cells were to come from some other patient or from some other source. uh, CRISPR-Cas9 can this machinery can also be put back into the patient by packaging these machinery in a friendly packet made of. There is some glitch that is happening.
Tuli, do you want to share your screen?
Yeah, just give me a second, please.
Mahima,
So
are you
sorry.
having troubles?
No, something is happening with the presentation. So anyways, Tuli, you can continue.
Yeah, just give me a second, please.
I have shared the screen.
Oh, okay, great. Yeah, so can you move to the next slide?
Yeah.
So instead of taking out cells from the patient's body, we can directly supply the CRISPR-Cas9 machinery enclosed within some friendly material like the lipid, which will not result in any immune response in them, and either put it back into circulation or introduce them directly into the target organ like liver. So CRISPR can also be used to detect genetic diseases. So what is currently being done is that scientists at UC Berkeley and at KGI Institute have come up with this chip, which looks like a glass slide, where basically the Cas9 and the guide RNA are stuck. So when we supply our sample DNA sequence to check whether any mutation is there or not, if there is a mutation, you can actually see this purple guide RNA binding to that region. And in this way, when this binding happens between the guide RNA and the DNA sequence that we have supplied to the chip, it actually leads in a voltage deflection. And this voltage deflection gives a beautiful message saying target sequence identified. So in this way, CRISPR can actually be used to detect genetic diseases from DNA samples. And while the rest of the world is in a spree to use CRISPR for various reasons, India isn't far behind. A group of scientists led by Debojyoti Chakraborty from CSIR IGIB have actually come up with a paper strip based test for detecting the COVID-19 infection. And this method is not only cost effective, it is also way faster than the current method that is being used in the labs for the detection of coronavirus infection. So as you can see here, if there is an infection in the sample, that if there are virus particles in the sample that we have provided, you will see two lines instead of one line. Okay, and this particular innovation is being commercialized with the help of Tata. So the group of scientists have from IGIB have a tie up with Tata, so that this test can get approved from the government and also be manufactured at large scale, so that you and I can use it easily. And now I hope you can appreciate what CRISPR can actually do. But there are things that have also come into the news about regarding this particular technique. One is the patent controversy. So there are many lawsuits going between these two groups. One led by Jennifer Doudna and Emmanuel Charpentier, who invented that CRISPR-Cas9 can be used to cut certain parts of the DNA, and the other group on the other side is led by Feng Zhang, who took this entire setup a step further so that they can be used in eukaryotic cells and in the cells of uh, humans and animals. So there is a legal battle between these two groups, so as to who gets the credit for this particular invention. But this is not the only concern around CRISPR. There is a major bioethical issue. You see, CRISPR can edit genome. So if something can edit anything, it is capable of changing it or altering it. So what actually CRISPR can do is to make designer babies. And what do I mean by designer babies? Well, since your genes may give you your traits, you can actually alter the traits that you want in a baby, right? So if you want a high IQ, you can do that. If you want the baby to be a sprinter, you can switch on or switch off genes that can achieve this. And there is a major concern among the in the scientific community as to the repercussions that such alterations in the embryo can have on the environment, on the ecosystem, because it might tilt the balance that is maintained in our ecosystem. But there is a silver lining. In a few articles that I read in this CRISPR journal, I took the liberty to pick up two quotes. The first one you can read, and what Henry Greeley is actually trying to say is that, hey, calm down. We currently do not have the information, or we do not know the genes that can help in increasing IQ, or that can give you stronger muscles so that you can run faster. So we still have time to pause ourselves and formulate rules so that we can regulate the judicious use of this particular genome editing technique and other scientific advances that come henceforth. And the second quote that I took the liberty to pick up is urging the scientific community to join hands with science policymakers and other experts and include their voices so that we can come up with a proper formulation and a proper guidance for how to use CRISPR-Cas9 in ways that can be used for social welfare. And on this note, I would like to end this talk. And my heartiest congratulations to Professor Dudna and Professor Charpentier for giving the human race this amazing technology. So we will now take questions.
Thanks, Tuli and Mahima. There's one question that we need to answer. How is CRISPR different than site-directed mutagenesis? or other technologies that were there before CRISPR? Why is CRISPR so revolutionary? Which part of that question?
Okay, that's a great question. Wait, I'll answer that. So other techniques, as in, okay, like site-directed mutagenesis, and all of these, they need enzymes like other nucleases. The basic idea is, the basic question that comes out from this question is that there were techniques previously, like nucleases, that are capable of cutting the nucleic acid or the DNA in our case. And why is CRISPR important? So if I call a nuclease a scissor, CRISPR is like a laser. Okay, so yeah. So if you take a look at this slide, there were restriction enzymes had been invented a long time back. In fact, the inventors of restriction enzymes actually got the Nobel Prize in 1978. So what CRISPR enzymes can do is, you're familiar with these palindromic sequences, right? So it can actually bind to these palindromic sequences, cut them, and this is how site-directed mutagenesis and all of this can happen. But the problem is the sequence that restriction enzymes recognize are short. So if you look at this code that you can see on the screen, and you search for a short code like cat, you will see in the genome there is a higher probability of finding this cat. So there are like 1, 2, 3, 4, 5, 6, 7, 8. So there are 8 places where you can see cat, cat, cat appearing. So other techniques will end up chopping up the DNA instead of precisely cutting at a particular point that we want. On the other hand, if we were using a longer sequence for identification, like cat, 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 you see that it appears only once. So the probability of a longer sequence to appear within a genome is lesser, and so we can hit the precision. And CRISPR can do that because the size of the guide RNA is pretty long. So it's about precision and it's about speed. Yeah. Are there any more questions, Farah?
Sorry, we'll just wait for a minute and just check if there are any more questions.
Yeah, I can see one. I think it's by Pradeep Chatterjee. If we can change, do you want to read out the question?
No. Carry on.
Yeah, if we change the DNA, isn't it going to affect the ecosystem equilibrium in future? Like I said, Mr. Chatterjee, yes, it can, and that is why it's a concern. It can skew the population in what we humans might think is desirable, but and that is why we need more and more voices like yours to actually come together and help the scientific community come up with a proper guidance manual.
Are there any more questions on FB Live? Can we check that?
No, I've been looking at Facebook and there are no questions as of now on Facebook, but we'll give it a minute anyway. And anything else on YouTube that we haven't taken?
No, basically comments which have been addressed, but no questions.
Okay, so in case there are no more questions, I would like to say that thanks a lot for
one has
this
popped
beautiful
up.
introduction into the world of CRISPR. I know it's a little bit technically challenging to try and summarize the entire development in this genetic cutting exercise in half an hour, and that too for a lay audience who really doesn't know much about the tools that have already been there and are in use. So thanks a lot. And it looks like this is an amazing thing, which not just designer babies, but it could be used for so many useful things for society. And on that note, I'd actually like to remind people that our last presentation on the Nobel Prizes for this year is going to be next weekend. And that's going to be the one on the black hole at the center of the Milky Way. So do join us next weekend also at 11 o'clock. For the second half of the Physics Nobel Prize. Till that time, stay safe and have a great week ahead of you. And thanks again, Tuli and Mahima, for this fantastic introduction to CRISPR. Okay, thank you.
Okay.